We're now going to talk about a reaction of alcohols that is a very practical reaction. It's widely used in certain circumstances in organic synthesis. It's this reaction where we make what's called a tosylate or sulfonate ester. An ester is a molecule where we have a carbon group and an oxygen attached to an acid in place of an OH. We've really only looked at esters of organic acids, what we actually technically call carboxylate esters. But we can make esters of other acids, other oxyacids that have OHs on them, like sulfuric acid, phosphoric acid, and so forth. In this particular case, we're going to be making an ester of what's called a sulfonic acid. Let's first look at our observed reaction and the abbreviations we're going to be using. You are totally welcome to use these abbreviations. All organic chemists do. We're going to start with an alcohol. We're going to treat it with this, tosyl chloride, TSCl. We're also going to put in a base called pyridine. We're not going to worry about the structure of pyridine right now. We'll be discussing that in a later chapter. Just understand that pyridine is a base. Its primary function is to absorb this hydrogen that we need to get out of the way so we can put the tosylate on there. At the end of the reaction, what we will see is that the hydrogen has been replaced with the tosyl group. We're going to see that these two letters represent a very large structure, but nevertheless, organic chemists are perfectly comfortable just writing these two letters. So let's look at what TS is. TS stands for para-toluene sulfonyl, which is a big word, but it basically looks like this. It's a benzene ring, has a methyl group on one side. Directly opposite that methyl group is going to be a sulfur with 2 bonds to oxygen. We write it generally as an expanded octet, sulfur double bond. If we wanted to, we could write single bonds and formal charges. Most people just write it this way. We can see then that this would be attached to something. In this case, it would be an oxygen and then a carbon. So therefore, that makes that an ester. The mechanism of this reaction, interestingly enough, is basically an SN2 mechanism. What we have is the oxygen is going to act like a nucleophile. The tosyl has an extremely large positive formal charge. It's very reactive with lone pairs. So a lone pair of the alcohol comes over, makes a bond to this sulfur of the tosyl group. There's a chlorine attached here. So as it makes that bond to sulfur, the chlorine is pushed off as chloride ion. So essentially we have a nucleophile coming in, on the opposite side, a chloride ion being pushed out. In fact, if we could monitor the three-dimensionality of this, we would see that it probably goes through backside attack and it probably features inversion. At this point then, we get this species. This is a really crazy looking species, but if we look, it's basically very similar to hydronium. Hydronium is an oxygen with 3 covalent bonds to hydrogen. In this case, what we have is an oxygen with 1 covalent bond to hydrogen, then a covalent bond to our tosyl right there on that sulfur, and then a covalent bond to our carbon group. That's going to give it a positive formal charge. It's also going to make it a reasonably strong acid. So now if we have a base, and pyridine is a base, the base can deprotonate this intermediate, leaving us with our tosylate. So now, why do we make tosylates? It's a pretty off-the-wall functional group. Well, it turns out that tosylates are really good leaving groups, similar to chlorines, similar to bromines, similar to iodines. Sometimes it's just going to be more convenient to make the tosylate and use it as a leaving group rather than trying to either use acid conditions and use the hydroxyl group directly or try to replace the hydroxyl group with a halogen leaving group. I'm going to show you an example. Imagine that we wanted to make this alkene. Last semester we talked about reactions that would make alkenes. We saw, for example, that we had a reaction where we could start with an alcohol where we would have an H here instead of the tosyl, and we could treat it with strong acid. In that case, we would protonate the alcohol, it would come off, we'd get a carbocation, we would remove a beta hydrogen, but we would remove the more substituted beta hydrogen, and we would actually not get this regioisomer, we would get that regioisomer. So the dehydration reaction wouldn't work. If we tried to use the primary, we would make a carbocation, but it would rearrange to secondary, it would then rearrange to tertiary, and again, we would get the wrong regioisomer. We could also, in theory, buy the halide here. But what if we didn't have access to that? This would be a really easy alcohol to make with a Grignard reagent. So what if we made our alcohol with the Grignard reagent, now we wanted to make our alkene? Well, what we could do is we could use tosylate. So we would convert our alcohol to this tosylate, and then what we can do is we can use a strong base and use this as a leaving group. This would be our alpha carbon, and then we would have a choice of betas. To get this beta that I chose, this is the less substituted beta, so we would use a sterically hindered strong base like T-butyl. So do you see, by using the tosylate, it's possibly easier to get us to our desired product than by either trying to use dehydration or by trying to convert this to a halide, which, by the way, is going to be the next reaction we talk about. That's why tosylates are used. They save us steps sometimes, and sometimes they're just the right solution. We're now going to look at reactions that convert alcohols to alkyl halides. This is kind of funny because one of the first reactions we looked at making alcohols was to start with an alkyl halide and make the alcohol. But this in many ways is how the organic chemistry science has been mapped out. We find a transformation in one direction, and then we look at if we can reverse that transformation. That gives us the possibility of making a wide variety of molecules. In this case, we're going to be able to replace that hydroxyl group with many different halides using several different reagents. I'm going to focus on the most commonly used reagents, and we're probably not going to go into great mechanistic detail here, but we are going to tell you some of the limitations of each, why we choose one over the other. Having said that, I'm going to tell you most of the time you're going to use PBr3, but nevertheless, they do have their strengths and weaknesses. Okay, first of all, we can use just hydrobromic acid, actually hydrogen bromide. I say hydrogen bromide because actually, although it's not written here, we generally use HBr gas, not HBr aqueous. We want to keep the water out. If we mix a molecule with HBr gas, we're going to replace the hydroxyl with a bromide ion. This is very versatile. It works on primary, secondary, and tertiary alcohols. However, its acid conditions, carbocations can be formed, and we do often see rearrangements. A primary alcohol is going to use an SN2-like mechanism. The acid protonates, makes a good leaving group. The bromide knocks off that leaving group as a water. The secondaries and tertiaries are going to be more of an SN1. We protonate, the alcohol comes off, we make a cation, it rearranges, and then we put the bromide on. We're not really going to focus so much on this aspect. We may put a question or two in the homework. But the main thing is, we need to get this on a flashcard and get this memorized. We can also use these reagents. These are phosphorus tribromide and phosphorus trichloride, and actually we can also use the phosphorus pentabromide and phosphorus pentachloride, although they're less commonly used for this. These are solids or liquids, and we mix them without a solvent, generally, with the alcohol itself. And what we see is that the phosphorus helps remove the oxygen, and then the bromide comes in and substitutes for it. It's basically an SN2-like mechanism. It really only works well on primaries and secondaries, although I am going to say with heat, it can also work on tertiaries and even on aromatics, although usually we go for different reagents for those. It does go by SN2 reaction, and we do observe inversion in both of these cases. Finally then, we're going to look at the basic workhorse of this reaction. SOCl2, which is called thionyl chloride. Thionyl chloride is a liquid reagent. When we want to use it, we just use it pure. We mix it with the alcohol directly. We often heat it a little bit. It works on primary, secondary, and tertiary alcohols. It gives off hydrogen chloride gas as a byproduct, so sometimes we have to make arrangements to capture that gas because it's very corrosive and toxic. But other than that, it's an excellent reagent. It's been used for over 100 years, and it works with a wide variety of hydroxyl groups. Particularly importantly, it works on hydroxyl groups on carboxylic acids, we're going to see later, which aren't even alcohols but can still be replaced with chlorines by thionyl chloride. For right now, we're just looking at alcohols. The really interesting thing about thionyl chloride is that it replaces alcohols on chirality centers with retention of configuration. So for example, if we look here, this hydroxyl group is on a wedge. If we treat it with thionyl chloride, the chloride product will also be on a wedge. It's super interesting. It's very useful for controlling stereochemistry, and it's a really widely used reagent. Finally then, I want to just briefly talk to you about how oxidation of organic molecules occurs in biological systems. Now, this is not the only way that molecules are oxidized, but it is an extremely common way. What we see is that alcohols, when they are oxidized, effectively we remove a hydrogen minus and a hydrogen plus. So we're removing hydrogen minus, hydrogen plus. If we imagine putting those two pieces together, we would have a molecule of hydrogen gas. In biological systems, many reactions are sort of controlled and catalyzed by what we call enzymes, which are very large protein-based molecules that catalyze chemical reactions. Enzymes generally have names, not always, but many of them have names that relate to how they change the structures of their substrates. The reactants that enzymes work with are called substrates. The names then would have, for example, a description of the structure change, and then they would end in ASE. So if we had an enzyme that was removing hydrogen, we would call that a dehydrogenase enzyme. Many people pronounce these dehydrogenase. So what we see is that dehydrogenase enzymes most definitely exist in biological systems. There's a wide number of them. Some of them work on a whole bunch of different alcohols, and some of them literally only work on one specific alcohol with one specific structure. Dehydrogenase enzymes don't work by themselves, however. They need another reactant. That reactant is actually going to be what removes the hydrogen minus. The reactant for a dehydrogenase enzyme, in most cases, is a molecule whose name is abbreviated NAD positive, NAD plus. These reactants are called enzyme cofactors. When you study biochemistry, you're going to learn a number of different enzymes reactions. You'll learn the names of the enzymes, you'll learn the structure of the substrates, and you will learn the cofactors. I'm going to just look at one of these right now. If we mix an alcohol with an appropriate dehydrogenase enzyme and an NAD plus cofactor, what we see is we remove this hydrogen, it becomes attached to the NAD, so because it's a hydrogen negative and the NAD is positive, we end up with an NAD with a hydrogen attached and no charge anymore. The two charges cancel out. We're also going to remove that as a proton, so we're going to get a hydrogen plus, although recall that in general we don't have hydrogen plus floating around in solution, so it's going to be attached to some type of Bronsted-Lowry base, and we'll talk a little bit about how that works. This is how biological oxidation reactions are written in biological chemistry. We have substrate, NAD plus, enzyme, product, NADH plus hydrogen plus. There are many different dehydrogenase enzymes. As I mentioned, some work on only specific alcohol substrates and some work on different ones. And typically, they kind of pair the dehydrogenase name with the name for the specific substrate or the name of the biological system. So, for example, in the citric acid or Krebs cycle, we have malate dehydrogenase, which, by the way, uses a different cofactor, but okay. And in other systems, we have things like horse liver alcohol dehydrogenase. All right. Now let's look at the cofactor really briefly. The cofactor is actually a dinucleotide. Nucleotides are the building blocks of DNA and RNA. This one happens to be an RNA-related one because it has both oxygens here. You can put them together in various ways to make multiples of these nucleotides. In this particular case, this half here is actually the A building block of DNA GCAT. So DNA, DNA is GCAT, RNA is GCAU. I take it back. This is the A building block of RNA, GCAU. It's the A. It's adenosine monophosphate, essentially. This nucleotide is one that's not used in DNA, but it has this base right here attached to our sugar piece. This is called nicotinamide. What we have here then is nicotinamide adenosine dinucleotide. Nicotinamide adenosine dinucleotide, NAD. It turns out, although we have this giant molecule here, the only part that's actually going to be reacting in the reaction is this base piece right here. This is the active portion, the nicotinamide. So I'm going to abbreviate the rest of it by just putting a squiggly line, and the rest of it would be there. The rest of it is probably used as a recognition element to get this piece into the place where it belongs. Here's then how that enzyme reaction works. We have our substrate, and the substrate fits into a space on the enzyme so that this hydrogen, or this hydrogen, depending on how we draw it, this hydrogen is aligned very close to that carbon right there of the nicotinamide. We then have over here a base from our enzyme, so something like an NH2 or the nitrogen of a proline, or there's various bases that are used in these things. That's going to be set up right next to the hydrogen that we want to remove. We can then look at this as kind of starting here, although it may start on this end. The base removes the hydrogen. This pair of electrons pushes down toward the carbon. That's going to try to push something out. It pushes out that bond. That bond comes over here and makes a bond to this carbon. Now, interesting, that carbon is willing to make a bond because it can push a pair of electrons out of the way here, and we can push in a pair of electrons onto the nitrogen, getting rid of the positive charge. So it's like that positive charge on the nitrogen is pulling that hydrogen toward it on the opposite side. At the end then, look at what we get. We get an aldehyde, or it could have two carbon groups. It could be a ketone. We get a protonated form, the conjugate acid of our original base. That's going to have to be removed. It might be removed by taking the aldehyde out, putting a water in, water removing that, water coming out as hydronium. It might be removed by another base and shuttled out and removed by water. There's lots of ways that we can get rid of that hydrogen to set us up to do another one. And then we have the NADH, and that comes out of the enzyme, goes into solution, and then is moved to various parts of the cell to do other things, the ultimate goal of which is to capture the pair of electrons here and to regenerate the NAD+, so we can do this again. So this is how a biochemical reaction works. I think what's really interesting about this is that if we look at this, this is an organic chemistry reaction. We're using the same type of things and drawing the same type of arrows that we draw in basic organic chemistry. This is one of the reasons why it's really strongly recommended that you get kind of a foundation in organic chemistry if you're going to study how biological systems function. Biochemistry, physiology, and even many branches of medicinal chemistry, in other words, chemistry of medicines and pharmaceuticals, depend on understanding the underlying organic chemistry. That concludes our lecture on this section. I just want to point out that in the notes we have a summary of all these reactions that you can use as a starting point for making your flashcards.